US, and his PhD from Rice University in biomedical engineering. Professor Missirlis was assistant and associate professor at McMaster University in Canada before joining the University of Patras in 1981 as a full professor, directing since then the Laboratory of Biomechanics and Biomedical Engineering until his retirement in 2013. He served as a vice rector of the university, as a member of the European and World Council of Biomechanics. He is an honorary member of European Society of Biomechanics and of ESB Biomaterials. In 2019, he was elected as a Fellow of Biomaterials Science and Engineering. He has co-authored a textbook called Biomaterials, A Tantalus Experience, co-edited 2 books, Modern Aspects of Protein Absorption on Biomaterials, and The Role of Platelets in Blood Biomaterial Interactions. He has published over 90 peer-reviewed papers in international journals, several book chapters in books, and currently is active in the area of cell-material interactions, mechanotransduction, tissue engineering, biomechanics from nano to macro level. Professor Missirlis is currently a Sunum affiliated researcher, as I have explained previously, and a collaborator of LTFN laboratory at Aristotle University in Thessaloniki, Greece. So we are very excited to have Yannis with us today, and I will give the microphone to you. You have the microphone.
Thank you, Fazilet, for this nice introduction, which was too long, but united. Good morning to all of you. As he said, I started as a chemical engineer, and Fazilet told me that chemical engineers can do everything. So I moved from that to now narrating stories about science, not so much data. If you are expecting fascinating data, probably you will be disappointed. But being retired officially, but still active, I have now a motto that comes from a very old grandfather of mine, saying that true wisdom comes to each of us when we realize how little we understand about life, ourselves, and the world around us. Now, probably you know that this guy Socrates never wrote a thing, and it is usually his students who said that he said that. But anyway, I follow this dictum in many ways. Now, usually I try to find ideas from mythology, history, that intermingle with science. So when I wrote with a good friend of mine, Joseph Helsen, a book called Biomaterials, we decided to put this subtitle, A Tantalus Experience. Now, I don't know how many of you know who Tantalus was. You will see in the later slide. But here is Tantalus in Karagol, which is near Izmir. And he did something that the system didn't like it. And you know, if you are against the system, sometimes you pay the consequences. So what the gods did to him, he was in this lake. Each time there was an apple tree, each time he was hungry, he tried to reach an apple, but the apples went up. Each time he wanted to drink, the level of water went down, so he was suffering all his life. So the idea was that biomaterials, everybody wants and claims that this is the perfect biomaterial for this application. It is a Tantalus experience. That is not a perfect biomaterial in any aspect. Anyway, Who was Tantalus? He was the son of Zeus and immortal. And in the introduction of Orestes, which is a tragedy by Euripides, Electra narrates the genealogy of the house of Atreus, where Agamemnon, I hope some of these words are familiar to you, starting with Tantalus. Now, if you see the periodic table of elements, in the same column, you find Niobium and Tantalum. And Niobe was the daughter of Tantalus. So, you see there are some connections. The Tantalum, now, the element, is very much used in biomaterials. And as you see in these recent titles, there is one called Preparation, Modifications, and Clinical Application of Porous Tantalum Scaffolds. And also there are many alloys of tantalum used in clinics nowadays. So even tantalum or tantalum is not perfect, so people try to use alloys to make it better and better. So, trying to talk about science, we have to have a methodology. So, what is a scientific method? How we conduct science? Is knowledge only scientific or sometimes intuitive? What is progress and what is development? I mean, the concepts of progress and development are not necessarily on the same track and on the same direction. There are standards, criteria, values, and or aims that we have to have clear when we try to conduct science. Now, another concept is quality of what we are doing versus progress, which is a result-oriented concept, while quality is an activity-oriented progress. So, even my kids sometimes, they question what is truth? Who knows what is truth? Or truth-likeness. Can we reach the goals we set up even in experiment we are conducting? Or these, some of those goals are utopian? I like also this statement that the thesis that science is progressive is an overall claim about scientific activities. It does not imply that each particular step in science has in fact been progressive. Individual scientists, like me and you, make mistakes, and even the scientific community is fallible in its collective judgments. So, but if we have a methodology and we set up clear our goals and how we go about it, then it is easier to find answers to such strong statements. Now, especially the young ones, when they try to do their thesis or later on, know very well things like citation index, impact factors, the so-called metrics, which try to ascertain the quality of their production in research. You know, take these statements from the Stanford University philosophy department called Plato, and here you see it is no doubt correct that one cannot advance scientific knowledge without influencing the epistemic state of the scientific community. But the impact of a publication as such only shows that it has moved the scientific community in some direction. If science is goal-directed, as we said before, then we must acknowledge that movement in the wrong direction does not constitute progress. So many times, and you will see hundreds of cases where people strive to produce and produce without really paying attention to their method. And here is an excerpt from some published data that show, for example, a few years ago, China's Ministry of Science and Technology found nearly 486 researchers guilty of participating in peer review fraud. University of Tokyo in 2017 determined that a very prominent researcher, Watanabe, tampered with images in 5 prominent publications. At the same month, another researcher in Tagawa requested that 3 of his published papers be withdrawn from the literature, bringing its total retractions to 17. There are many such stories, but look at that, the last one. In October 2017, the expert group on scientific misconduct at Sweden's Central Ethics Review Board, it's a huge title, but you can see the importance of that if it works well, studied a case that the magazine Scientist produced, that of surgeon Paolo Macchiarini, who was previously been found guilty of misconduct regarding the synthetic trachea transplantations she conducted that led to the death of at least 3 patients. So there are many such things, and also that year, 19 editorial board members of Scientific Reports, which is a prestigious journal, resigned from the journal over a study that was allegedly plagiarized, but the journal refused to retract. So there are many cases of fraud. That's the message. I will skip that because it will take me some time. So why I said this as an introduction, to show that science and publications and results shouldn't be taken on granted without examining them. And science progresses. But we don't know if that progress is real. We know what is real according to the technology that we have at our hands at a specific period of time that can give us measurable results, data. If technology progresses, we found out later on that the theory or the story or the model that we had as true is not true anymore. So I will take these 5 examples, the Froman effect, I will explain that for those who don't know, the brain-gut connections, the immune system and the brain, mechanics in life sciences, and some thoughts on neural transmission. Now, this guy here, I don't know how many of you know, is the so-called father of biomaterials, Alan Hoffman. Unfortunately, last month he died in Seattle, and he is really the father. And we had a meeting, you will see later on protein adsorption on biomaterials, pathogens, and we try to ascertain the Froman effect. What is the Froman effect? Froman was another scientist from Brookings. It is a highly dynamic protein adsorption process on biomaterials. So the idea in the 60s was that the total amount of absorbed proteins remains constant, but identity of proteins change over time. Actually, first, it is supported, if you go to the literature in the 60s, early 70s, you will see that if you put a biomaterial in blood or in blood equivalent solution, first albumin will absorb, then it will be displaced by IgG, then fibrinogen, then factor 12, high molecular weight kininogen are sequentially absorbed, one displacing the other. So that was the dogma on protein material interactions at the time. So when we had this workshop 30 almost years later, and we published a book about the protein absorption of biomaterials, the whole thing changed because the people there showed that it's not only 5 proteins. There are hundreds of proteins that fight for a space to attach on a biomaterial. And later on, in 2017, through new technology that is also using nanoparticles and proteomics, more than 300 proteins were detected at all point times. So it's not only that they fight and they display, but more than 300 are there and they stick and they fight. So the whole concept of a Frohman effect in the 60s, because technology advanced somehow, then some people were able to detect in different concentrations a whole total or more number of proteins. And this is important when you use, for example, filters for catharsis and things like that. So, if you put a biomaterial as an artificial organ to implant it, there are tissue damage, the Frohman effect, and in the end, there are possible outcomes depending on the type of material. These are degradable, these are non-degradable, and then you see that encapsulation, for example, here is important, tissue integration, and foreign body reaction. Foreign body reaction is always present, but here in the degradable ones, you form a fibrous scar instead of encapsulation. Now, let's move to the defensive system. In the brain and the central nervous system, we have microglia. We have the meninges that carry cerebrospinal fluid, but also lymphatic vessels. There is the blood-brain barrier and cytokines. So these are what we know today as members of the defensive system of the central nervous system. Now, in the rest of the body, as all of you know, I'm sure, the immune system is divided between the innate and the adaptive. This is the immediate reaction, fast and blood, go and try to kill anything that they find. And then the adaptive, which is slow and focused, mainly involving the lymphocytes. Neuroimmunology now, so that means the combining of the two is more, focused, because peripheral immune cells make an integrin that allows them to penetrate the blood-brain barrier, which for many years was thought to be exclusive for the entrance of immune cells into the brain. So this story comes out a few years ago from a guy called Kipnis, which shows that lymphatic vessels release a lot of cytokines and they allow peripheral immune cells to enter the brain. Now, again, I want you to notice that any cartoon, either static or dynamic, gives you only a glimpse of the information, because this is a very dynamic system, and all I'm trying here, or the researchers who did it, trying to show you concepts, not really what is happening and what are the dynamics of, how to say, fight between the cells, the matrices and all that. In that sense, also this researcher talked about the 7th sense. So apart from the 6 known senses, you know, vision, touch, hearing, taste, smell, and balance, there is this system that includes immune cells, cytokines, but also microbes that probably, there is this thesis put forward that this is the 7th sense of our bodies. And talking about microbes, let's see what are these guys. How the microbiome challenges our concept of self. The sense of self, who we are, I mean our body, includes the adaptive immune system, the brain, the genome, but the human is a dynamic and interactive community of human and microbial cells. You cannot say that these are two separate things. It is interactive and dynamic from birth until the end of a life. The composition of the gut microbiome determines the profile of some cell populations. Some have anti-inflammatory functions and others pro-inflammatory action. So what we have mainly in our gut microbiota determines many functions of our existence in a crucial way. The gut microbiome produces short-chain fatty acids that differentiate beta cells to plasma cells to secrete either protective IgAs and anti-allergic effects. The evidence that microbiome is a major player in neurodevelopment and neurodegenerative diseases amounts day after day. So, the microbiome is not influencing the nuclear genome. It is co-constituting the meta-organisms that we are. So this is a concept that if you don't follow it, then you have two different systems and you say one influences the other. No. The new ideas, which not everybody agrees with them, is that the interaction is continuous. Here? Okay, it's better now. Sorry. So it's a new concept. The initial one was this. This is who we are. Now, defining selfhood in the modern way, or at least until now, is that you have the brain, you have the immunity and the genome, and all this is an interactive system. So everybody knows that the brain and the gut, through the vagus nerve, talk to each other all the time. And there are many efferent and afferent routes where information is going up and down. And many clinical disorders, like psychiatric disorders or inflammatory GI disorders, all of them are a product of this interaction and depends what information comes out of here to the brain and back, and what information comes from here up to go up there depends on the amount and the constitution of the microbiota that we have in the gut. Okay, so in this axis, there is a complex interplay between mechanical influences. It is the first time I'm talking mechanics. You know, I'm a guru for mechanics in life science. I believe very much in that, so you will see some of this later on. So it is mechanics, hormones, gut bacteria, bile acids, and your neurocircuitry. I was fascinated to learn that 90% of the body's serotonin is produced in the gut. Part relies on microbes. So microbes in the gut, specific ones, produce serotonin, which is a neurotransmitter. Obesity, sorry, I'm one of the obese guys here, is a disease of the gut-brain axis. And now there are, instead of pharmaceuticals, these people call them electroceuticals, there are chemicals from the neural system called spleen neurotransmitters, which may regulate the immune system. And human trials with those electroceuticals are now on trying to relieve people who have rheumatoid arthritis, Crohn's disease, lupus, etc. Even there is ultrasound stimulation of US stimulation of spleen nerve reduces inflammatory arthritis. And that was a few years ago a good publication on that. Now, let's move into another part. And by the way, if you want to stop me and ask a question while I'm talking, it will be good for me because I can drink some water and so, coming back to these biomaterials when we insert it in the body, again, I present a cartoon here, and it is representative, but pay the attention that is needed, not more attention to that. So, if this is a cell, and this diagram is a surface, so the cell that goes onto the surface, it tries to find out what is there and attaches it. And these are the focal adhesions. And inside there is the cytoskeleton of the cell and there is the nucleus. Okay, if we try to enlarge one of these focal adhesions, we see it here. So this is the membrane of the cell, those two black lines, the extracellular matrix or the biomaterial, or the extracellular matrix on top of the biomaterial. And we see some biomolecules, including the family of integrins that come from inside and go outside to interrogate what is there. And when we say interrogate, it's not only chemistry, it's not only to find the chemical composition. They try to find electricity, is there some change in current? Mechanics, is there a small earthquake there? What is happening? And they connect to many other biomolecules in this area, like focal adhesion kinase, vinculin, paxillin, tallin, and so on and so forth. And the current dogma in biology is that whatever information the integrins bring from outside the cell, through the membrane, inside, again, emphasize, whatever information comes in, through biochemical reactions with this family that exists here, goes all the way, these reactions change the biomolecules, and it goes all the way to the nucleus. Now, there is this microfilaments and the cytoskeletal fibers, which could be microtubules, actin, and intermediate filaments and other things, also connect whatever happens here to the nucleus. And some people, including myself, say, or we claim, that if there is a mechanical signal down here, goes directly through the microtubules and the microfilaments into the nucleus without having a very, how to say, important interrogation with biochemistry. Now, this is a strong statement, so I'm trying to repeat it again. There is information that there is a biochemical message that goes there. There is now some information that mechanical message goes there, and on the way, both of them are interacting and affecting the speed of transmission, both the biochemical one and the mechanical one. Now, if we think this is right, instead of 3 or 4 or 10 elements, you remember here? An analysis a few years ago showed that the complexity is huge, just in one or in thousands of focal adhesions. So suppose you are a true researcher, you know, those people who really want to find out things. And you learn that in that focal adhesion, you have all those guys or girls or it or whatever, which connect to each other. What does it mean, connect? They give information, they take information, they integrate information. And then you are determined to study it. How we study it? Usually, the golden law is we keep everything else constant and we change one parameter. Then 2 parameters, etc. Now, if you want to study this, it will take you 2 lifetimes. And it will be very, very expensive. And then some people say, oh, okay, let me take this one or that one because they are in the center. So then we have to decide why evolution brought, for example, this one in the end that has only 3 interacting. Actin has 2 men. Some others have 5. Which is more important? Or is there redundancy in our biology? So these are questions that, at least I don't know anybody answering them in a convincing way. So if I go back, but I will go forward here, to talk about, okay, this is like, I'm sorry, it's a second year university lesson, but I think it's very important. The comparison between biochemical and mechanical signals. Because if we don't understand that, it is difficult to ascertain how to carry out experiments, say in epigenetics, in the expression of genes, etc. Now, chemical signals all over outside here or inside our bodies propagate through diffusion of molecules in the cytoplasm, and their speed is limited by the chemical reaction rates, for example, phosphorylation, the binding or the diffusion rates. If somebody disagrees with that, let's talk about it. So it's diffusion. But diffusion there is a simple law of diffusion, and depends a lot on the diffusion coefficient, which is of this order. You know, these are 4 orders of magnitude difference. And in all, it says that a molecule needs about from 1 to 100 seconds to travel 10 microns by diffusion. So, let's say 10 seconds to go a distance of 10 microns. Let's see now the mechanical signals. Mechanical signals are transmitted through the deformation of the cytoskeleton, that is the actin myosin, microtubules, etc., along actin filaments, microtubules, and intermediate filaments. So these filaments are deformed. Now, the speed of the mechanical signals depends on the elasticity of the cytoskeleton. They travel the same distance, 10 microns, in sub-second timescales. So they are much faster. Mechanical signals travel faster than chemical ones. Is there a reason for that? Or what is the effect if we take those two and forget about electrical and other signals? Just comparing the two. Mechanical signals can be transmitted over long distances and broad areas and to different organelles through the network of elastic actin cytoskeleton. For example, from the nucleus mesprine, plasma membrane, proteins, mitochondria, etc., etc. So again, the picture is that information from outside, biochemical and mechanical, move all to the head of the cell, that is the brain, that is the chromatin and DNA. But the mechanics go there first. Why they go there first? We'll see in a moment. But that is important. This guy Grinnell published 30 years ago in Science and showed that the motor nerve terminals release neurotransmitters within 10 milliseconds after cell surface integrins are mechanically stressed. Excellent experiment. He did that in Dallas. But this good publication died because the main dogma in biology doesn't care about mechanics much. They get cell cultures and everything. The most they can do is, without knowing the forces, they shake them, and then they see biochemistry mainly. Now, if we include both, and already somebody pressed this button, yes, it's me, we can now understand what could be the reason that mechanics go there first. So, again, a cartoon, don't believe it 100%, just get a glimpse of that. So this is the membrane, extracellular matrix. There is stiffness outside, tension and torsion, because we are all very viscoelastic entities. So the information, as I said before, comes inside. There is biochemical information coming here, but also there is mechanical information, and they go all the way to the chromatin. Now, a few years ago, I had this opinion paper published that said the following: a cell, this one, in contact with extracellular matrix and with a neighboring cell, and under fluid conditions, receives information and reacts to it either by ion channel openings, by junction proteins, by integrins, etc. And all this information in the milieu in cytoplasm and in the cytoskeleton integrates itself, goes through the link complex and through lamins to chromatins. Now imagine if the mechanical signal goes to chromatin, what it does is to open it up, to stress it or unstress it, depends on its convolution at that moment. So that the biochemical signals, which are also, or even they do the final act, so their importance is undoubtedly important, they couldn't enter inside if the chromatin is like, how you call this word, when people knit, you know, they have all this thing inside them, you cannot enter. So mechanics are important to facilitate the information, biological information that comes into the nucleus, so that it will either do its epigenetics plus or minus. Yeah. Sorry that my, I don't know, the autonomous system presses that. I move now into another one, to me very fascinating. But what this author, Fox, said, you can read in the title, that hardware limitations influence discoveries scientists made and which ideas enter mainstream scientific thought. So, to have a scientific thought enter into our brains depends on the hardware limitations of having data that supports this idea. If we cannot measure something which is there, but we cannot measure it because we don't have an AFM in 1965, then we cannot really talk about it. So there was this guy, Ishiki Tasaki, was first to publish, before Second World War, that the action potential jump from gap to gap in the myelin sheath around some axons. 1939, who knew about it? It was Japan. Japan was entering the war. Nobody knew about it. Then, what? 13 years later, it was the Hodgkin and Huxley, using electrodes, that they proposed the currently accepted model of action potential transmission, the ionic flows, for which 10 years later they got a Nobel. Now, Tasaki and others observed, and now some people continued his research, that during the firing of a pulse on a neuron, the fiber axon briefly widened, then narrowed again, and the this thickness change was only 7 nanometers. Couldn't measure it. But also the temperature was increased and then fell back again with the number of Celsius was 10 to the minus 6. And he claimed that the pulse, the action potential, is a mechanical one. This guy Snyder, in 2014, working with artificial membranes, confirmed the all or none phenomenon of a voltage pulse and a mechanical pulse simultaneously. Heimburg said that anesthetics work by changing the mechanical properties of the nerve, because anesthetics are huge difference in the molecules, so it couldn't be only ionic flows. The proposed model then of these guys in a few years ago was that a mechanical shock wave, as it advances, squeezes lipid molecules to liquid crystals, there is a phase change that releases a bit of heat. So again, they don't claim that there are not ionic flows, but they claim that the mechanical event during this communication is very, very important, and it facilitates the other things that are happening. Nocta, tell me when I have to stop, because I can talk for hours. So I will skip that. This is the picture of the ideas. For example, you see this lipid membranes, and then becomes a crystal, and then again and again, but it's too much information. Look, there was a publication in 2005 by Heimburg that said, on soliton propagation in biomembranes and nerves. Now, it was published in a very prestigious proceedings of National Academy. You know how many citations it has? 25. They just dumped it. Then, they found another way to increase attendance to this idea. In 2015, in Nature Communications, these people said the mechanical surface waves accompany action potential propagation. You know, they put water into the wine to try to convince the community that something is there. And finally, 4 years ago, Snyder and his colleagues said it sounds is a mechanical, like an action potential, unification of electrical, chemical, and mechanical aspects of acoustic pulses in liquids. Now, sometimes I challenge my audience. How many biologists know about that? Or neuroscientists? I am now having an office in neurophysiology department there. I asked the prof, his postdocs, nobody cares about it because they have the experiments, they have the hypothalamus, they have the hippocampus, and they do the experiments, this and that on a mouse, etc., etc. We have to widen our views about things. Now there is new information where memory is finding itself. So most attention has been given to synapses, to alter transmission, to use drugs, or to check neurological diseases. But millions of neurons in different brain areas must be linked to produce coherent memory. Of course, it's not only neurons. It's not only neurons. There are glial cells that respond to neural activity. They sense neurotransmitters released during firing. So a neurotransmitter is not confined in synapses. That's what I learned when I was studying it. But all along the axons. And facilitate, that is glial cells, more myelin production. Then comes the astrocytes that control the amount of neurotransmitter in the nodes of Ranvier, using thrombin to thin myelin. Now, I thought thrombin is only a hematological agent. But no, it plays, and this is all around biology and physiology, that a particular player has, or rather plays in many, many different levels, and sometimes contradicting levels. So myelin, you know, is crucial for optimal information transmission, but with age, and I can ascertain to that, something is happening more to my myelin than yours. So, people start thinking about thrombin. Is it a friend or a foe? Inflammation and coagulation, they have interconnected pathways. So nerve injury increases thrombin levels. When you have low thrombin levels, these Schwann cells release factors to extend neurites. And when you have high thrombin levels, it inhibits the work of Schwann cells, release leading to demyelination of paraneurons. So you can see that it's not a friend or a foe, it's both a friend or a foe depending on the conditions. So thrombin is everywhere. Thrombin and neurological disease and injury, it involves in stroke, in infections, epilepsy, Alzheimer's, you name it. So from something that we knew before, that it is in the cascade of coagulation, it seems it's very important in many neurological diseases as well as functions. Okay, now this is really challenging because it says that our memory depends on mechanics. Memory is stored, according to Gould, who is now expanding a lot now, is stored in the brain in a mechanically encoded binary format, written into the conformations of proteins found in the cell ECM adhesions. You remember the ones I told you? That organize each and every synapse. So he claims that every synapse of the billions, trillions of synapses we have play a role in our memory in a mechanical way, usually or especially involving tallinn. Now, tallinn contains 13 binary switches and exists in all synapses. The synaptic signals trigger the cytoskeleton to push and pull on these switches. The cytoskeleton serves as the levers and gears that perform the calculation. And key to this, maintaining the mechanical balance of the cell, are the 20 members of the Rho family of GTPases, which have 80 activators and 70 deactivators of these proteins. So she goes to very much detail on, and there is another guy, he was at Columbia, Michael Siege, that all his life worked on the mechanics of tallinn. Very good work. And he ended up in Singapore, where he continued his work. Now, the mechanical layer integrates with the chemical and ionic ones to process and store information. So, according to him, every synapse can store information. The synapse is a complex computational device. Okay, I will skip that because it shows something that will take us a lot of time. But I was happy, you know, because Greek, a neighbor, I mean, my grandfather was from Ocak, but, you know, a friend of mine, Erdis, says that I am 50% Greek and 50% Turk, or an Aegean person, or whatever you want to call it. But there were ancient computers, and maybe you have heard the Antikythera mechanism that was built in 2nd century BC, and this is how they found it in underwater, sunken ship. And when they reconstruct it, it is considered worldwide the first mechanical computer. So to me, I said, why not memory, computation, and all that, not to have a mechanical element? Why? because life starts with mechanics. We are on Earth. We are not in space. If one of these guys, spermatozoarium, doesn't know fluid mechanics, swimming biomechanics, impact mechanics, fusion mechanics, and penetration mechanics, life doesn't start. We live on Earth. There is G. You are sitting. You are not flowing. So mechanics is important, and then maybe evolution used it a lot in how we have built up ourselves. Also, I'll skip that because it's a lot of information. Even, you remember I talked about this memory. There is a very recent article a few months ago called Progress of the Italian in the Mechanical Basis Theory of Memory, where they say that memory loss in certain diseases such as Alzheimer's or pathological conditions such as aging, lack of oxygen, may be associated with abnormal folding of tallinn, which is a mechanosensitive protein. So some people get on the track of these ideas and try to find details about what is happening. There are mechanically activated ion channels. I mean, all our ionic channels are activated by mechanics. Even exosomes, maybe you've heard about these guys everywhere. Now, they found out, especially in cancers, that the cancer cell exosome microRNA can transmit the mechanical force of where this cancer cell was sitting in the extracellular matrix to distant cells and participate in the construction of the extracellular microenvironment necessary for cancer cell metastasis. So these guys in cancer propagation, they don't only send away biochemical signals, but the memory of the mechanics where they originated. I mean, to me this is fascinating. I don't know how to study it, but some of you will do. Just to show that we are very complex things.
The cytoskeleton is a dynamic network of filamentous proteins that extends throughout the cytoplasm, forming the struts, cables, and girders that give the cell its shape, internal organization, and mechanical support for movement. There are several types of protein filament, each built from subunits that rapidly come together or break apart, allowing these elaborate structures to be assembled and dismantled wherever they're required by the cell. Microtubules are stiff, hollow tubes that anchor other organelles and serve as tracks that guide the movement of vesicles and other cell components. Actin are thin, flexible filaments that form cross-linked bundles and branching networks. Actin is particularly important for cell movement. They are central to the contractile engine in muscle cells and provide the mechanical force for cell movement at the plasma membrane.
So, why I show you this movie is to see the complexity of things happening in one cell, and we have trillions of cells. And it's good always to remember numbers when we do some studies. So, our somatic cells are about 15, 10 to the 12. Now, bacteria is much more, and viruses, many more. So this is the system that we are. So who is serving who? These guys came first, we came second. Some of them are bad guys, some good guys, etc. But also some of our cells are good guys and bad guys, cancers and all that. So it is a very complex thing that continues all the time. And then again, look at the numbers. So many neurons in the adult brain. Now, if you are a physical science person going into biology, remember that the typical relative viscosity in the cell, in the cytoplasm, is 5 or 6. Our cytoplasm is not water solution. It is more like a gel, 6 times as viscous as water. Now that has implications on movements and all that. Number of cell divisions, 10 to 16. Blood cell turnover, 110 to the 9th every day. So this superorganism with human and bacterial cells, along with viruses, cohabit, antagonize, fight, or protect each other. Now, if we don't have a sense of numbers and what each element is doing, it's difficult to conduct thought experiments at least. Okay, skip that also. A new journal. I was so happy because Xu Qian was a friend of mine, and it started it 3 months ago. It's called Mechanobiology in Medicine. And in his first article, Xu Qian from La Jolla in California says that mechanobiology is a multidisciplinary research field that studies how cells perceive and respond to mechanical signals to regulate their biological function, such as development, proliferation, remodeling, movement, and migration. I couldn't put it as nice as he did. And this signal transduction includes molecular, genetic, and epigenetic mechanisms, as I proposed a few years ago. Things change. Who has seen this movie called The Graduate? Long time ago. Yeah, she's white hair, so she knows that. So, I put 1 minute of it, so you can.
Your future, your life.
Well, that's a little
Dustin
hard to say.
Hoffman.
Ben, excuse me. Mr. McGuire.
Ben,
Mr. McGuire.
come with me for a minute. I want to talk to you. Excuse us, Joanne? Yes. Thank you.
That was in the 60s.
I just want to say one word to you. Just one word.
Yes, sir.
Are you listening?
Yes, sir.
Plastics.
It was the era of plastics, because they are produced plastics. Now, 2 out of 3 documentaries worldwide say how bad plastics is for nature, for turtles, for everything, for our own, in Greece. Now they investigate drinking water from these plastic boxes. There are many nanoparticles of plastic, plasticides that we imbibe all the time. So I finished some future thoughts. There is interconnectivity of things. So reductionism, which we are doing all our lives in science, we have to be very, very careful. If you act here, there is a reaction there. Attempt to solve a problem, look out for creating another one. Physics gives us some important information of its subatomic particles, and the characteristics of nucleons arises out of collective behavior of quarks and gluons, which is an emergent phenomenon. similarities with biological processes. I must pay homage. This is a meeting, it says here, 10 years ago in Koysegis, whatever, yes. And this is a bunch of old biomaterial people. Unfortunately, Alan, as I said before, died last month, and Julio also passed away in Madrid a few months ago. The rest of us, I don't know who will go first, but we were a good bunch of people. And in the end, we need an environment to do whatever I proposed. So, apart from science, let's try to have this. Thank you.
We can take a couple of questions. Is there any?
Thank you for your very interesting and thought-provoking talk. Lots of things to go and read and think about. But I wanted to ask a little bit about the mechanoepigenetics concept that you shared. So I guess the common dogma is that keeping chromatin open is about the methylations and other chemical, other acetylations, other treatments on the chromatin. So are you saying that the mechanics opens the chromatin to allow that to happen?
Yes.
Okay.
Yes, that's the basic concept. If you ask me, have you done an experiment to prove it? No.
Yeah.
I have not done. But people are thinking of doing it.
Okay. Yeah. Thank you. That's what I thought. That's good to hear.
I don't think that one. I have a very strong voice, but okay, I'll use. First, I apologize. I'm neither a biologist nor am I anywhere close. I'm a mechanical engineer doing material science here. It was a wonderful talk. Wonderful. I mean, I learned so much. Just about in one of your slides, there was this very important link between mechanical force and temperature. Basically, temperature is mechanical force. Delta T equals stress.
Dynamics is everywhere.
So that actually brings one more additional complication. That is, so it's very difficult to separate strain, especially strain rate, from diffusion, because it creates temperature and then it changes the diffusion properties. Yeah, so that was, although there's maybe a solution that going up and down I'm going cold and warm, or that could actually really change the diffusion, or separate the two, diffusion from the two. Right. That was one thing, and it was shown there. It was really nice. And then the other thing I forgot, because I think I'm the only one who actually remembers the graduate one. Yes, now I remember. Of course, in order to mechanical force the strain to be transmitted at the speeds that was mentioned here, basically it's the speed of sound. But this requires primary bolts, and that requires only along a chain. So that means the information is only transmitted in one direction, or along the chain of the molecule. Because from chain to chain it's secondary bonds, and then it becomes very viscous, and then it will damp out rather rapidly.
It's a very good question, this one. Just a couple of years ago, people started studying what is the rate of making an actin fiber such that the sound, the mechanics, move along it. And sometimes the rate of making a microfilament of actin, or microtubule, or intermediate filaments is similar to the rate of the mechanical signal that is transmitted along. Now, it's not a fiber. It is an interconnected, how to say, a scheme of so many fibers of different chemical composition. Some cross the nuclear envelope, some stop before. So the speed of mechanical information inside the nucleus, because there is a nucleoplasm there with higher viscosity than the cytoplasm, change also. Now, all these are more or less theoretical aspects. We see the beginning and the end. We don't know the process. So it needs a lot, that's what I'm saying. We don't know many things. So how the rate of mechanical signal along acting or even along microtubule, we don't know. Is it double? Is it the same? Does it changes as the loop between a biochemical signal that affects whether the, you remember I showed you the destruction of a microtubule, this, and the coming back. Now this is not obvious how it happens. It needs initiators, it needs catalysts, it needs so many things. Recruitment of molecules from far away, it's a very complex. So what I liked is that I'm provoking your thought. If you like this concept, okay, let's try to see how we are going to study this. If we think it's important, some people might say, oh, this is rubbish. But if you think it's important, then if you sit down and make a very nice analysis of how to learn about it, how to study, how to measure, then maybe some progress will be in sight.
Do you have questions?
Thank you very much for your time. It was a great talk.
Thank you.
Thank you.